My name is Bill Catterall. I'm a professor of pharmacology at the University of Washington in Seattle. And today I'd like to present a second lecture in a series of 3 on electrical signaling as part of the iBiology series. I'm very pleased to participate in this series, which puts together science in a way that is accessible to many people. So in this second lecture in the series, I'll say just a few words about electrical signaling processes at the beginning, and then move quickly to recent research which has shown us how voltage-gated sodium channels, the key molecules in electrical signaling, work at the atomic level. So this slide illustrates how action potentials or electrical signals are generated in a nerve or muscle cell from the classic work of Hodgkin and Huxley in the 1950s. They studied the squid giant axon, and they recorded the action potential in a squid giant axon as shown here in the trace labeled EM. The action potential begins with the resting membrane potential here at about minus 65 millivolts. The cell is depolarized all the way to plus 20 millivolts or higher, and then the action potential returns back to the baseline here at the resting membrane potential. This electrical signal is how nerve cells communicate with each other and transmit information to muscles and other tissues. It's also the way that nerve cells process information in the brain. And electrical signals control many other fast processes in biology. The action potential is generated by the coordinate action of 2 types of ion channels. Voltage-gated sodium channels, which are responsible for the rising phase of the action potential here. And sodium channels really do 2 things. They activate, which causes the rising phase of the action potential, and then they inactivate here, bringing the sodium current back to 0 within a millisecond or 2. On a slower time scale, potassium channels activate, and they allow potassium to leave the cell, and therefore they repolarize the cell membrane back to the resting level. The sodium and calcium channels, sodium and potassium channels, are 2 members of a large family of ion channel proteins. This slide illustrates the molecular relationships, the ancestral relationships among the different ion channels that we find in the human genome. There are 143 related ion channel proteins in the human genome, and the sodium channels were the founding members of this large family of signaling proteins. Here in the blue quadrant, you see the relationships among 10 different sodium and calcium channel genes in the human genome. The sodium channels are here. Pardon me, the sodium channels are here in blue. Calcium channels are just next to them. On the left side of this dendrogram, you can see the voltage-gated potassium channels. There are 70 of them in the human genome. Other channels called trip channels are shown in green and yellow. And there are 10 different sodium channels and 10 different calcium channels encoded in the human genome. So we'll focus on sodium channels primarily, but I want to point out while I have this slide that sodium channels and calcium channels arise from a common ancestor. Here at the base of this dendrogram, there is an ancestral protein that gave rise to all 20 sodium and calcium channels that we find in the human genome through evolutionary diversification. And we think this molecule is a bacterial sodium channel, which we will study using X-ray crystallography and gain insights at the atomic level into how sodium channels and calcium channels work. So this slide illustrates the difference between a vertebrate sodium channel and a bacterial sodium channel. On the left part of the slide is illustrated a vertebrate sodium channel. As you can see, it contains 24 transmembrane segments organized in 4 homologous domains. Each homologous domain contains 6 transmembrane segments. within those 6 transmembrane segments, there are 2 functionally distinct modules. There's a voltage sensing module here, the S1 through S4 segments in each domain, and a pore forming module, highlighted in green, the S5 and S6 segments in each domain. This is a very complicated macromolecule. In addition to all of its 24 transmembrane segments, it has very large intracellular domains, as illustrated by these long loops. And so this is a very difficult protein to study by structural biology methods, even by the most modern structural biology methods. So a key discovery in our field was made by David Clapham, whose name is here on the publications noted on the bottom of the slide. So he and his colleagues discovered the first of the bacterial sodium channels, illustrated here, and the first one they called NaChBac. And as you can see, NaChBac has the structure of one of the 4 homologous domains of a vertebrate sodium or calcium channel, and it doesn't have any of the large intracellular or extracellular loops. So it's a very simple, minimalist protein containing only a voltage sensor and a pore-forming domain. So we think then this is the perfect target for structural studies of sodium channels so that we can understand the atomic level basis for their function. So we isolated several new bacterial channels, and we were fortunate to find one that worked well in biochemical expression and purification, formation of crystals, and finally in X-ray crystallography. This image shows the bacterial sodium channel that we study, which is called NavAb, for voltage-gated sodium channel from Archobacter butzleri. And for most of the images that I'll show you, we're studying NavAb into which we have inserted one single amino acid mutation. Isoleucine number 217 was changed to cysteine, or I217C, as you see here on the slide. This gave us higher resolution of our X-ray crystallographic images and greatly facilitated determining the structure of the channel. So here in the image, you're looking down on the sodium channel, and you can see its pore in the middle through which sodium ions would move, and it's surrounded in blue by 4 pore-forming modules. And then on the periphery of the structure, in the warmer colors, are the 4 voltage sensors. And you can see that they are approximately symmetrically arranged around the center part of the molecule formed by the pore module. So this shows the 2 functionally distinct aspects of sodium channels in clear relief: the pore modules in the center and the voltage sensing modules on the periphery. So here you can see the sodium channel on its side, so as you would see it from the side in the membrane. And here in this drawing, the voltage sensing modules are in green. You can see there are 4 of them. The pore module is in blue in the center, and the connecting linker, called the S4-S5 linker, is in red. And this illustrates the functional separation of the different parts of the channel. And as we'll see later, when voltage drives a conformational change in the voltage sensing modules, they pull on the S4-S5 linker, and that pulling motion opens the pore and allows sodium to move through. So my talk is organized in a sequence of events that take place as the sodium channel functions. So first, we'll talk about voltage sensing, which is carried out by the voltage sensing modules, of course. So here is the structure of the NavAb voltage sensor at 2.7 angstroms resolution as we found it in our X-ray crystal structures. It's superimposed on a voltage sensor of a potassium channel studied in a different lab in the field, in Rod McKinnon's lab at Rockefeller University. And they're superimposed to illustrate the structural similarity of the voltage sensors between these two different types of voltage-gated ion channels, and makes the point that sodium and potassium channels really are very similar in the way that they sense voltage. So looking at this voltage sensing module then, you recall that it contains 4 transmembrane segments numbered S1 through S4. And here in the drawing, you see S1 on the left over here, and then S2, S3, and S4. A key part of this drawing is the illustration of positively charged amino acid residues in transmembrane segment S4. These are called the gating charges. These positively charged residues are the part of the voltage sensor that feels the electric field. So the positive charge is in the electric field across the membrane, and as the membrane potential changes, an electrical force is exerted on those positive charges, and they move, initiating the conformational change that eventually opens the pore. So the 4 gating charges are all arginine residues, or R, in NavAb, and they're illustrated in yellow writing here, and you can see the side chain of R1, R2, R3, and R4 illustrated in the drawing. Now this drawing also has 3 key landmarks in the voltage sensor that are necessary to understand how it works. So here on the outer or extracellular side of the voltage sensor is the extracellular negative cluster, or ENC. And it's illustrated by 2 red side chains of negatively charged amino acids right here. And they're interacting closely with the gating charges in the S4 segment and neutralizing those positive charges by interaction with the negative charges. A second key location in the voltage sensor, a functional component, is the hydrophobic constriction site, shown in green just here, HCS. And it's composed of hydrophobic residues in the middle of the voltage sensor that work as a hydrophobic seal, as kind of a rubber seal that prevent the movement of water and ions from outside to inside through the voltage sensor. Finally, on the intracellular side of the hydrophobic constriction site, and again illustrated in red, is the intracellular negative cluster, and it's composed again of negatively charged amino acid residues whose side chains are highlighted in red just here. And again, they're interacting with gating charges. They're interacting with the R4 gating charge here of NavAb. So the way that we think the voltage sensor works is when the membrane potential changes, the voltage sensor changes conformation in a way that's driven by the electrostatic force on the gating charges. And the gating charges move out and back in, and as they do, they exchange ion pair partners from the intracellular negative cluster through the hydrophobic constriction site to the extracellular negative cluster. So the gating charges are always neutralized by interaction with negatively charged amino acid residues, but as the voltage sensor moves out under the influence of depolarization, it exchanges ion pair partners from the intracellular negative cluster to the extracellular negative cluster. And you'll see that in a movie as we go along. So this drawing is a 2-state movie, but the illustration that you see here is the resting state of the voltage sensor. So here in the resting state, the channel is closed right at this point. This is the gate that closes the channel pore. And the voltage sensor is in its resting or deactivated conformation. You can judge that because the gating charges, illustrated here by the blue balls right there are drawn inward toward the inside of the cell. Upon depolarization, there's an electrical force on those gating charges, as I explained on the previous slide, and they'll move outward like this. That movement will cause a pulling on this linker, and the linker will pull open the gate of the channel and allow sodium movement. So now I'm going to run this little movie, and you'll see those events take place, and the movie will come back several times during the talk. So if you don't catch all the details of the movement the first time, you'll have 2 or 3 more opportunities to do that. So here we go. There, the gating charges moved out, pulled on the linker, and opened the pore. So that movie is really just a model of how the sodium channel works. And actually, I'm going to go back to it just one more time. So it makes a number of predictions that we can test experimentally. So the way we test those predictions is we select 2 amino acids that are far apart in the resting state of the channel, but come close together in the activated state, according to our channel model. And we substitute those amino acid residues with the amino acid cysteine. Cysteine is a sulfhydryl-containing amino acid, and it's chemically reactive. Cysteines react with each other to form disulfide bonds in proteins. So it's important to know that the bacterial sodium channels, including NavAb, are cysteine-free. They have no cysteines of their own. So when we put in 2 cysteines, if they form a disulfide bond, we know it's being formed by the cysteines that we put in, not by any native cysteines present in the structure. So the experiments on the next slide then, we put cysteines in the place of the gating charges in different positions in the S4 segment, and we ask, do they interact with a negative charge, this one out here, in the resting state or in the activated state? The experimental results from that kind of experiment are shown on this slide. Now, this is an electrophysiology experiment, so I'll kind of go through it step by step. So here, with the R1 gating charge mutated to cysteine, and a charge in the negative cluster, E43 mutated to cysteine, when we try to measure the sodium current in the resting state of the cells, we see no sodium current, as illustrated by the black trace that's labeled control. So those 2 cysteines are forming a disulfide bond and locking up the voltage sensor in the resting state. So we see no sodium current when we start the experiment. But we can reduce the disulfide bond with a sulfhydryl reagent, beta-mercaptoethanol or beta-ME on these slides. As we do that, the sodium channel wakes up and you get a sodium current, as shown by the green trace here. And down below, you can see for the various black traces that the wild type, the single mutants, all work fine. They give sodium currents throughout the experiment, but the double cysteine mutant gives no sodium current at the beginning of the experiment, here at the bottom, and only when we perfuse in the beta-mercaptoethanol do we see sodium current. So this tells us the gating charge R1 and the extracellular negative cluster residue, E43, are near each other in the resting state, but not in the activated state. In the center, we've done the same thing with the gating charge R2. So R2 is partially interacting with E43 in the resting state. When we stimulate the cells and put them into the activated state, we lose that sodium current that we started with. And then as we perfuse in the beta-mercaptoethanol, the sodium current returns. So these results say that R2 is partially interacting with E43 in the resting state, but not fully. Then on this right side of the slide, same type of experiment, but now it's with R3 and E43. And with R3, we get the full sodium current at the beginning of the experiment. So R3 and E43 do not interact with each other in the resting state, but as we depolarize the cell, we progressively lose the sodium current so that that interaction is made in the activated state of the voltage sensor. And we can recover the sodium current by adding beta-mercaptoethanol to reduce the disulfide bonds and return the channel to normal function. So these 3 experiments are illustrative of a whole series of experiments of this kind in which we have mapped the interactions of each of the gating charges in the voltage sensor with each of the members of the extracellular and intracellular negative clusters. And in that way, we have fully confirmed the movie that I showed you previously, which illustrates how sodium channels are activated by voltage. And so here's that movie again. And we'll run it in just a moment. But again, you should watch as the our 1, 2, 3, and 4 gating charges move out and then pull on the linker and open the pore. This type of function of the voltage sensor has been studied by many groups, and until recently there'd been quite a bit of controversy about that. But now, based on our work and others, there's an emerging consensus on voltage-dependent gating mechanisms that this is the way that the voltage sensor works. And so this voltage sensing function really is at the core of all electrical signaling in biology. So once the voltage sensor activates, the next step is the pore needs to open. So the next couple of slides will show you how we think the pore opens. So first on the left, from our X-ray crystallography, you can see the architecture of the sodium channel pore. The gray part here shows the water-filled area that sodium ions would move through from outside the cell to inside. On either side of that gray area are shown the pore lining S5, S5, and S6 segments from 2 subunits. And there would be a 3rd subunit behind the plane of the slide and a 4th in front of the plane of the slide in a real sodium channel. And the slide illustrates the functional areas of the pore. An extracellular funnel where sodium enters, a narrow ion selectivity filter where sodium is selected relative to other ions, a central cavity which is water-filled, and an activation gate at the intracellular end. In our crystal structure, the activation gate is tightly closed, as you see here, occluded by the side chains of methionine 221 located right at the base of the S6 segment. The pore is closed, but as I showed on the previous slide, the voltage sensors are activated because 3 of the gating charges have moved out in our crystal structure of NavAb. This means that the channel is in a pre-open state, a natural state in the functional cycle of the channel in which the voltage sensor has been activated, but the pore has not yet opened. In a normal channel, this state would only last for less than a millisecond. But remember, we did our crystal structure on a mutant, and we think that mutation has trapped the channel in the pre-open state. If our channel is in the pre-open state, then it's interesting to compare its structures with other channels that have been crystallized in an open state in order to see the transition that takes place upon opening. And one such channel is the Kv1.2 potassium channel studied by the lab of Rod McKinnon. So this shows the transition for the voltage sensor and the pore that takes place during opening by comparing the structures of the pre-open sodium channel and the open potassium channel. And you can see here that opening involves a rolling motion of the voltage sensors kind of around this way as the S4 segment moves out, as you have seen. That exerts a torque on the S4, S5 linker, sort of in the plane of the membrane like this. That in turn exerts a torque on these pore lining residues, and they open the pore with kind of a twisting iris-like motion, just like the iris in your camera. So now we come back to our same movie, but I want you to focus here. This is where the activation gate is. This is where the channel opens. So when we run the movie this time, the voltage sensor will move again. It will pull on this linker again, and watch the subtle rotation movement of these transmembrane segments right here that opens the pore like a camera iris. And just with that brief opening then, there's a pulse of sodium current, and that depolarizes the cell and begins electrical signaling. Now this slide is also a movie. It's just the same as the previous one, except that we've added an inactivation gate. Vertebrate sodium channels have a fast inactivation gate associated with them. And here we've added that just as an illustration to the model of the bacterial sodium channel to show you what would happen during fast inactivation of the type of sodium channel that Hodgkin and Huxley first studied when they discovered voltage-dependent activation and fast inactivation. And as you'll see when we run this movie, exactly the same as the previous one, except that the inactivation gate will close and shut off the movement of sodium ions. Like that. So that's the kind of sodium channel movement that takes place during every action potential in nerve or muscle in a vertebrate. So let's go now to the next topic, which is slow inactivation. So as I mentioned, Hodgkin and Huxley discovered the process of fast sodium channel inactivation, which takes place in milliseconds. But there is a second inactivation process called slow inactivation. The bacterial channels don't have the molecular component needed for fast inactivation. On the previous slide, we pasted it in from a vertebrate sodium channel to illustrate. But the bacterial channels do have slow inactivation. So we can study the structural basis for slow inactivation by studying the bacterial channels. The left side of this slide illustrates the slow inactivation process of the bacterial channels. So during a single stimulation, bacterial channels open and then their current declines. And remember now that sodium current increases in the downward direction. So it's activation downward and then inactivation as it goes back, as the line goes back up. Moreover, if you repetitively stimulate these bacterial channels, the sodium current gets progressively smaller, depending on how fast you stimulate. If you stimulate more rapidly, the current declines essentially to zero. So this slow inactivation process in the bacterial channel is very powerful. It can completely shut down the channel, and at the same time, it's very slowly reversible. Now, this is the first slide I've shown you in which we are studying the wild type version of the bacterial channel NavAb, not the mutant I217C. And the reason we're studying the wild type is that the wild type undergoes the slow inactivation process, which seems to have been prevented in the mutation, which locked the channel in the pre-open state. So the wild type channel crystallized in a different conformation, and we believe that different conformation is the slow inactivated state of the bacterial sodium channel. And that's illustrated here. You can see changes between at the level of the selectivity filter, which is essentially square in the pre-open state, but moves here to look more like a rectangle or a parallelogram in structure in the inactivated state. At the central cavity, you can see again that the channel has gone from a square symmetric conformation in the pre-open state to a more parallelogram conformation in the slow inactivated state. And then at the bottom of the pore, at the activation gate, there's a symmetrical arrangement of the intracellular ends of the S4 segments in the pre-open state, the S6 segments in the pre-open state, whereas it's oblong or oval and asymmetric in the slow inactivated state. This panel perhaps shows best that the change in structure involves the inward movement toward the pore axis of two of the pore lining S6 segments, and the outward movement away from the central axis of the other two, creating a very asymmetric structure compared to the symmetric structure of the pre-open state. So We think then that slow inactivation is caused by this asymmetric pore collapse, which takes place during prolonged or repetitive depolarization. This slow inactivation process in nerve or muscle is important to protect the cells from too much excitability, and it's also important to determine the frequency and length of trains of action potentials that might be generated in those cells. So our last topic is sodium conductance and selectivity, and of course it's an important topic. Sodium channels wouldn't be sodium channels if they couldn't select and conduct sodium very efficiently. So again, on the left of this slide, you see the architecture of the pore with its different components, and then the right side focuses on the selectivity filter. And remember, that's the narrow part of the pore. So here you are on the right, looking down on the selectivity filter as a sodium ion would as it enters the pore. And the key point is that this part of the selectivity filter is surrounded by 4 negatively charged glutamate side chains, one on each side. So it's a very highly charged location, 4 negative charges in a small space, and we call this the high field strength site because of that. This square or almost square arrangement of the glutamate residues in this high field strength site is kept in place by 4 hydrogen bonds shown by dotted lines as here, one on each corner of the square, which are inter-subunit hydrogen bonds between a tryptophan in one subunit and a threonine in its neighbor, and they act as staples to hold the ion selectivity filter in its active configuration. Now the left side of this slide again illustrates the ion selectivity filter at its outer end, at the high field strength site, but then on the right, the new image shows, looking from the side, the arrangement of the other sites that sodium would interact with as it enters and passes through the ion selectivity filter. So the outermost site is the high field strength site, as we've seen before. It's formed by glutamate 177. Then there's a central site formed by the backbone carbonyls of leucine 176, the next amino acid in line. And then on the inner side of the selectivity filter, there's a third site formed by the backbone carbonyl of threonine 175. And we believe that sodium interacts with each of these sites in sequence as it moves through the pore. And the size of the selectivity filter, approximately 4.6 angstroms in diameter, says that sodium moves through as a hydrated cation because sodium is too small to touch the sides of the selectivity filter if it moved through alone as a bare ion. So sodium keeps its waters of hydration as it moves inward through the pore. This is another view of the ion selectivity filter. On the left side, the ion selectivity filter is shown embedded in the rest of the channel molecule, so you can see where it fits into the overall structure. On the right side, this is a small movie which shows the parts of the selectivity filter that interact with sodium. The carboxylate anions of glutamate 177, the backbone carbonyl of leucine 176, and the backbone carbonyl of threonine 175 are all illustrated in red. And now this is again a little movie, and it will go around to give you a 3-dimensional sense of what sodium would see as it moves through the ion selectivity filter. So you can see the sequence of interactions between sodium and these oxygen ligands, either carboxyl or carbonyl groups. So potassium channels and sodium channels conduct their ions in very different ways. Here on the slide is illustrated the ion selectivity filter of a potassium channel in blue, just here, and it fits inside of the ion selectivity filter of the sodium channel, illustrated in yellow. It's much narrower, and that's because the potassium channel conducts bare potassium ions with no waters attached to them. The potassium ions interact directly with this series of backbone carbonyls as they go through the pore and into the cell. The sodium channel has a bigger ion selectivity filter because it conducts hydrated sodium ions, and it needs to be wider to allow sodium plus water to move through and make these specific interactions as it moves into the cell. We've investigated the binding sites that sodium visits along its way through the ion selectivity filter in two ways. In this slide, we've used the X-ray crystallography technique to directly image the sodium ions. And you can see that sodium ions are present in the outer vestibule, the blue balls here, the high field strength site in the ion selectivity filter, and in a site between the central and inner coordination sites, and again in a hydrated form in the central cavity. So we believe this diagram shows you again the pathway that sodium moves through, in this case shown by imaging sodium ions in the X-ray crystal structure. In this last slide, we've also looked at where sodium binds in a different way. We've used a technique of molecular dynamics which simulates the movement of sodium ions through the sodium channel using the chemical information from our channel structure and from the knowledge of sodium movement in solution. We've used molecular dynamics in an unbiased sampling mode, and we've recorded a relatively long time, 26 microseconds of sodium movement, which is enough time to allow hundreds of sodium ions to move in and out of the pore as we study it. And our studies show, studies done in collaboration with Professor Regis Pomus at the University of Toronto, our studies show that sodium spends most of its time in two sites, in the high field strength site here in green, and in a site between the high field strength site and the leucine backbone carbonyls here illustrated in yellow. In addition, we see sodium ions at low probability in the outer vestibule, just this low signal along this line, and in the central cavity here, this brown signal along this line. And so these data fit well with the X-ray crystallography data and indicate that sodium ions visit primarily two sites in the ion selectivity filter as they move from outside on the left in this drawing through the ion selectivity filter into the central cavity on the right. The bottom shows the chemical mechanism of sodium interaction with the sodium channel and the selectivity filter. So we believe that the selectivity filter is most stable when it has two ions in it, as here. When a third ion enters the selectivity filter, it bounces one of the others out, in this case into the central cavity and into the cell here, returning the selectivity filter to an occupancy of 2. Selectivity filter then can lose a sodium ion and go back and at low probability be occupied by only one sodium ion, but very quickly it picks up a second and goes back to the more stable 2 sodium bound state. And you can see that going through this cycle many times would effectively transport sodium from its high concentration outside the cell into the cell at lower concentrations. Okay, so with that, we have gone through the full cycle of sodium channel function at the atomic level, and I hope that's given you a view of how electrical signaling works from the perspective of the single sodium channel molecule that works to initiate all of the electrical signals in nerve, muscle, and other fast signaling tissues. Thank you for your attention.